Hello students, my name is Dr. P. Shashikiran. I am working as lecturer in botany at SKP Government Degree College, Guntakal. Today's topic that we'll be discussing is about plasma membrane, its structure and functions. So plasma membrane, you can say, call it as plasma lemma. The other name for plasma membrane or cell membrane is plasma lemma. So the objectives of today's lesson are: one is the plasma membrane structure, another one is plasma membrane models that were proposed. There are two models, one is the sandwich model and the fluid mosaic model. Third one is the plasma membrane functions. So here in this diagram figure you can see the cell membrane, that is plasma membrane, both in an eukaryotic cell and a prokaryotic cell, where the cell membrane is the layer or wall that surrounds the cytoplasm along with the other cell organelles that are present inside a cell, either in a eukaryotic cell or a prokaryotic cell. So this cell membrane that lies below the cell wall, here you can see. So outside this the excuse me, cell wall is there, the outermost layer. Below the cell wall you find the cell membrane or plasma membrane that surrounds the total protoplasm. Excuse me. So if you see plasma lemma or plasma membrane, so these are in both plants and animals. The cytoplasm is surrounded by a layer of wall that are known as plasma membrane or plasma lemma. The limiting membrane of the cell organelles like the mitochondria, Golgi complex, endoplasmic reticulum, nucleus or nuclear membrane, etc., and so on, are known as cell membrane. And the cell membrane and the plasma membrane are collectively known as biological membranes. If you look at the composition of cell membrane, so these are primarily made up of three important components. One is the protein, second one is the lipids, third one is the carbohydrates. Protein comprises the major share, that is, it comprises of around 60 to 80% of the total cell membrane composition, which are further classified into three types of proteins. One are the structural proteins, second one are the functional proteins, third one are the carrier proteins. Second important component present in the cell membrane are the lipids. So lipids comprise the second major share, comprising of around 20 to 40%, which include the phospholipids and the cholesterol. The third major component are the carbohydrates, which comprise of around 2 to 10%. Those include the glycolipids and the glycoproteins. So these are the important components that are included in the composition of a cell membrane. So the plasma membrane are generally semi-permeable in nature. You can say that these are selectively permeable, which are made up of phospholipid bilayer. So the so here in this diagram you can see, so this is a phospholipid, and here there are two parts you can see. One is the head region and another one is the tail region. So the head region of a phospholipid are hydrophilic in nature, and these are made up of the phosphate head, you can say, phosphate and glycerol. So these are generally hydrophilic in nature, which attract the water. And the tail region, you can say, that these are hydrophobic in nature, which are made up of fatty acids, that is the saturated and unsaturated. So the tails are generally hydrophobic, that is they repel the water. So they are not attracting the water, or that they are just repelling the water. Whereas the hydrophilic nature are seen in the head region, which are hydrophilic. So this is the basic structure of a phospholipid. So the structure of cell membrane or plasma membrane has been described by two models, which were there, and that are the sandwich model, and another one is the fluid mosaic model. The first one, if you see the sandwich model, it is also known as trilamellar model, which was proposed by Danieli and Davison in the year 1935. They studied the this plasma membrane in the starfish egg membrane from the point of its surface tension. They proposed that the two layers of lipid molecules were associated with a monomolecular layer of a polar globular protein molecules. So the electron microscopic picture of plasma membrane revealed a three-layer structure of that two layers were around 2.5 nm thickness and separated by a 4 nm space. So here in this picture, this here you can see this is the sandwich model proposed by Danieli and Davison model, that is the 1935 the year they proposed this model. Here you can see these are the phospholipid bilayer, two layers, where the head region is there and the inner tail region is there, and above that we find all these globular structures are nothing but the globular proteins. So here one more picture is there. Here you can see the membrane proteins, the above globular structure that are there, that is the these are the proteins, whereas the this region is the phospholipid bilayer. So this is one layer and this is another layer, and exterior to that we find the proteins, membrane proteins surrounded in both the layers. So this whole thing looks like a sandwich where the two slices of bread are there and inside that stuffing is there. So that's why this was named as sandwich model or trilamellar model. Further, another model was proposed by J.S.J. Singer and J.L. Nicholson in the year 1972, which was named as fluid mosaic model. They stated that the lipid molecules are present in a fluid state capable of rotating, moving laterally within the membrane, and the proteins occur as mosaic of discontinuous particles that penetrate deeply into the lipid sheet. So if you look at this picture, you can see that this whole thing, phospholipid bilayer, is nothing but just as a seabed, and in which you find globular proteins which are there as in a mosaic sort of pattern, just as icebergs, just they are floating on inside a sea. Okay, so just like that, if you see these iceberg-like structures that are floating in on a seabed, same way these floating structures are nothing but the globular proteins which are embedded in the sea of the phospholipid bilayer. So the pattern was just like a mosaic, hence this was known as fluid mosaic model. The fluid was nothing but the phospholipid bilayer, and the mosaic is nothing but the globular proteins. So in this picture you can see here there are glycoproteins. So the proteins with carbohydrate attached to them, And here glycolipids are there, the lipids with carbohydrate attached to them. And here you can see different protein channels. Channel proteins are there. Then peripheral proteins are there. Peripheral membrane proteins. Then the integral membrane proteins are there. And these cytoskeleton filaments are distributed below the plasma membrane. And here and there are cholesterol structures that are embedded in the phospholipid bilayer. So this whole thing is nothing but the phospholipid bilayer. So the cholesterol here, whatever these structures are there, these structures, they give the both rigidity and flexibility to the plasma membrane. Whereas there are two types of protein majorly, that is the extrinsic proteins. So these are known as extrinsic proteins or peripheral proteins, which are also aquaporins. They transport primarily the water. Then the intrinsic proteins are there, which these are the intrinsic proteins, which are known as integral membrane proteins, which help in the passive transport of ions or metabolites where no energy is utilized, so no ATPs are used. And so this type of transport of ions and metabolites are nothing but the passive transport. Whereas in active transport, the flow of the ion or metabolite is always against the concentration gradient, where definitely energy is required or utilized in the form of ATPs like the sodium and potassium pumps. Another thing is that another transport across the membrane is the cytosis, which is also known as vesicular transport, where two types are there, endocytosis and exocytosis. In endocytosis, what happens here is the either the solid or liquid material, these are engulfed through the plasma membrane inside the cell. So example is phagocytosis, pinocytosis, and so on. Whereas the exocytosis are example the neurotransmitters. So the signals are sent outside the cell. So this is an example of exocytosis where the material or signal is sent outside the from inner inside to outside the cell, that is the neurotransmitter is the example for that. So lastly, if you look at the functions of plasma membrane, excuse me. So first thing is that it keeps the all the cells intact. So it gives a mechanical support by keeping all these select cells that are present in the tissue collectively intact. Second thing is that it gives, it protects, it is a protective and barrier where the plasma membrane protects the inner components or cell organelles or the main protoplasm, including all cell organelles, from the attack of pathogen or any toxic material. Second, thirdly, it regulates the transportation either from inside to outside or outside to inside the cell. That's why it is selectively permeable or semi-permeable. So it does not allow everything inside the cell, or it does not let everything go outside the cell. So these are selectively permeable only with the help of these specialized proteins. The whatever material has to get transported either from inside to outside or outside to inside the cell. Another one is this, these are small lipid soluble molecules, that is the oxygen and carbon dioxide. These can pass easily through the plasma membrane, and water can freely cross across the membrane as the phospholipids are the hydrogen or hydrophilic in nature. So this is all about the uh, functions of plasma membrane. So to summarize, we discussed about the structure of plasma membrane, the composition of plasma membrane, then the two models that were proposed, that is the sandwich model and the which was proposed by Davison and Danelli, and the fluid mosaic model which was proposed by Singer and Nicholson. So the fluid mosaic model is the latest model of the plasma membrane that is mostly been accepted. And lastly, we discussed about the functions of plasma membrane. So that's all about plasma membrane structure and functions. Thank you.